Hello, the title of the work that I'm going to present is Subcortical Anatomy of the Ventral and Dorsal Attention Networks. Attention is a core cognitive function that undertakes the filtering and selection of relevant information by the brain. Two macroscale networks regulate attentional mechanisms. The ventral attention network, in orange, is a stimulus-driven system, while the dorsal attention network, in blue, mediates goal-driven processes. The study of the neuroanatomy of these networks has taken a corticocentric approach. However, some evidence suggests that they may also have subcortical components. First, electrophysiological recordings in primates have shown that subcortical structures such as the pulvinar and the superior colliculi also regulate attentional mechanisms. Second, the study of phylogenetic distant species with markedly different cortical morphologies show behavioral similarities with us during attention tasks. Finally, in humans, neglect, a paradigmatic syndrome of hemispatial inattention, does not only result from cortical dysfunction, but also from subcortical lesions. The alignment of individual functional maps exclusively based on structural features may lead to a misalignment of small subcortical nuclei. In a previous work, we have shown that if we perform an additional step of functional alignment, we may improve the identification of these small structures in average group analysis. Therefore, with this work, our aim was to explore the subcortical anatomy of the ventral and dorsal attention networks by the functional alignment of resting state functional MRI maps. Our hypothesis was that some subcortical nuclei, such as the pulvinar, striatum, superior colliculi, and locus coeruleus, also integrate these networks. To achieve this, first we computed the ventral and dorsal attention network maps by the synthetic analysis of 110 7 Tesla resting state functional MRI maps from the Human Connectome project. The seeding was performed from the Gordon's et al. Parcellation of cortex, in which one is composed by 23 cortical parcels and then by 32 cortical parcels. Then we performed the functional alignment of these maps with an iterative diffeomorphic transformation with ends. Group effects were analyzed with one sample permutation test. Third, we performed a connectivity analysis. For structural connectivity, we used a whole brain deterministic stratigraphy. For functional connectivity, we calculated partial correlations, and we also performed a graph theory analysis in which we analyzed the hub properties of network nodes. Finally, we analyzed the relationship of these maps with the neurotransmitter system by calculating the spatial correlation of the van and then brainstem nuclei projections with an atlas of 9 neurotransmitter systems provided by Hansen and colleagues. Here are the first results of our analysis, namely the cortical and cerebral surface projections of van and then maps, in which we can see the typical distribution of these networks along the cortical surface, as well as the different cerebellar projections. When we analyze the subcortical distribution of these maps, it is clear that some subcortical nuclei, such as the pulvinar, the superior colliculi, and also a cluster of other brainstem nuclei, such as the pedunculopontine system and the Raphe nuclei, also make part of these networks. Actually, a conjunction analysis showed that most of these nuclei are shared by both networks. In terms of connectivity, the structural connectivity analysis of Van showed that it is not only composed by the typical ventral association fibers, namely the third branch of the superior longitudinal fasciculus and the uncinate, but also by projection fibers, namely the frontopulvinar and the tetopulvinar fibers. In then, the structural connectivity map was also not only composed by the typical dorsal association fibers, namely the superior longitudinal fasciculus, first branch, but also by projections fibers, namely the frontopulvinar, the parietopulvinar, and tetopulvinar fibers. In graph theory analysis, we showed that these subcortical nuclei have high levels or high values of degree and betweenness centrality, showing that these nodes not only belong to the network, but they might also have a core role in their organization. Finally, the correlation analysis with neurotransmitter systems showed that Van and Dan were specially correlated with the distribution of alpha-4 beta-2 acetylcholine nicotine receptors, and also with the distribution of dopamine and serotonin transporters. With these results, we propose an improved model of the ventral attention network and dorsal attention network that includes the pulvinar, the superior colliculi, the caudate, and a cluster of brainstem nuclei. And also that the projections of these networks are correlated with the distribution of the acetylcholine alpha-4 beta-2 nicotine receptors, and also with the dopamine and serotonin transporters. The main limitation of our work is that with resting state analysis, we are unable to untangle the dynamic interactions that occur between these two networks, but future investigations with task-related fMRI in humans or direct electrical recording in animals using our model may answer this question. The fact that the alpha-4 beta-2 nicotine agonists and also dopamine transporter modulators such as methylphenidate and modafinil have established roles in attention modulation, increase the plausibility of our neuroanatomical neurochemical model. And we believe that this model may reconcile behavior data from distant phylogenetic species and also may contribute to a better understanding of attentional disorders in humans. Thank you very much.